Inside the Keim Lab, world-class research is being conducted year-round, working with collaborators from around the world.
So Joe, I hear we've got a great crop of undergraduates this year.
Well, we've got 23 undergrads right now. It's great.
You know, one of the things that I really like is the way we bring them in waves so that the older undergraduates and more experienced ones have a chance to train the other ones.
That's a critical part.
Joseph Bush began working with Paul Keim when he was a master's student 20 years ago. After earning his PhD at Purdue, he returned and works as the assistant laboratory director in the Center for Microbial Genetics and Genomics.
We do a lot of genetics. We're in the genomics age now, so we also do a lot of bacterial genomics, where we're sequencing every base pair in the genome of a pathogen, something like anthrax or plague or Burkholderia.
This lab provided key information about origins of anthrax used in the post-9/11 attacks. Many samples studied here are provided by scientific collaborators in other parts of the world. Studying DNA reveals a lot about a pathogen, which ultimately can keep populations safer.
A lot of these pathogens are diseases of wildlife, but they can also affect human beings. So we use our genetic techniques to look to see which pathogens are different and which ones are more similar. And DNA will tell you that, which is amazing.
I am focused mostly on Pasteurella multocida, which is a bacteria that causes avian cholera. So that's my main focus. I also do work on other projects such as white nose fungus, which is Geomyces destructans.
While everyone in this lab is required to learn many safety measures, Sabrina Herman is one of the few undergrads with the distinction of working in the biosafety cabinet. Her work is contributing to understanding of H5N1, or the bird flu.
I was able to track that the bacteria was traveling through migration patterns. So that alone is really important, just to not just assume, oh, it's here and then it's in Canada tomorrow. It's because of the migration patterns and because of the vectors that hold this bacteria. And we're able to kind of piece together a story and make sense of it.
There is a lot still to be discovered in these fields. Senior Lauren Gates works with the pathogen Coxiella burnetii, which causes Q fever. It affects livestock and humans too.
Oftentimes it causes an acute form of the disease, which is a lot like the flu. You're sick for about 10 days, you go home, you don't know that you even had Q fever. But in rare cases it'll cause a chronic form of the disease, which causes things as serious as endocarditis and even death.
Here in the Keim Lab, Gates studies DNA of the bacteria found in cow's milk. By understanding how the pathogen travels through landscapes and survives in different conditions, an outbreak could be prevented or stopped in its tracks.
If you realize, okay, there's this herd of cattle that's infected with Q fever, Coxiella, then you can go there and stop trade between other farms, and then that protects other animals and people coming in contact with these animals from acquiring the disease.
Gates is one of 23 undergraduates working at the Center for Microbial Genetics and Genomics, which employs 50 people who do critical research.
We're facing some important biological challenges on this planet, and a disease that could be easily spread is one of those challenges. And so spread by aerosol, or an aerosolized type of bioweapon, of course, is a major concern.
Work done here on the campus of Northern Arizona University has become increasingly imperative as people and wildlife travel the globe and quickly spread pathogens.